Hello, dear student in the third preparatory stage. In the last session, we talk about the cells in our bodies, their types, their division, and the stages for mitotic division, and the mitotic division result. In this session, we will discuss the meiotic cell division. Meiosis or the meiotic cell division. What's meant by meiosis? Meiosis is a kind of cell division, occurs in reproductive cells only, and it leads to the formation of male and female gametes and transferring of genetic traits from parents to their offspring. This division mainly for the reproductive cells in the body of animal or human or in the plant cell, mainly for the reproductive cells in all living organisms. The aim of this division, number one, formation of gametes, which is the male gamete and the female gamete. Also, transferring of the genetic traits from the mother and father to their offspring. The gametes in human and animals, the male gamete is called sperm, and the female gametes is called ovum. But in the plant cells, the male gametes is called the anther pollen grains, but in the female gamete is called the ovum. Each cell division begins and starts with interphase. The interphase is the stage of cell division that the cell prepares itself for the division through important biological processes. Also, it duplicates its genetic material and prepares itself for the other stages of the division. The phases of meiosis takes place in two stages. The first is first meiotic division and the second meiotic division. Firstly, we will talk about the first meiotic division. What is the aim of the first meiotic division? The first meiotic division formed to reduce the number of chromosomes in the cell nucleus to half its number to produce the gametes. So, the cell undergo the first meiotic division stages to decrease its chromosomes number into half for formation of gametes. The first stage in the first meiotic division is the prophase 1. What does the cell undergo in this stage? Number one, chromatin reticulum intensifies and condenses and appear in the form of distinct chromosomes. So the chromosomes appear. Then the chromosomes are arranged in homologous pairs. Each pair consists of 4 chromatids, so it is called tetrad. Each 2 homologous chromosomes in the tetrad move away from each other. Each chromosome consists of 2 chromatids linked together by the centromere. Also, the centrosome migrates to the cell pole to produce spindle fibers. The spindle fibers appears and the chromosomes connected to the spindle fibers. The nuclear membrane disappears and the tetrad floating on the surface of the cytoplasm without any membrane. Then the cell go to the metaphase 1. During this stage, the tetrads or the chromosomes pairs arranged at the cell equator or arranged at the cell center. Then the cell undergo anaphase 1. In this stage, every two homologous chromosomes move away from each other as the spindle fiber shrink. When the spindle fiber shrink, each tetrad splits into two separated chromosomes. Each chromosome moves to the cell pool. One of the two chromosomes migrates towards a cell pool and the other migrates toward the other pool. Therefore, each pool contains half the number of chromosomes of the parent cell. The last stage in this first meiotic division is the telophase 1. In this stage, the nuclear membrane is formed around the chromosomes to form the nucleus, which surrounds half the number of chromosomes in each pool, leading to the formation of two nuclei. Each nucleus contains half the original number of chromosomes of the parent cell. So each cell contains n chromosome. After this phase, which is the telophase 1, the cell will enter into the second meiotic division. In the second meiotic division, it aims to increase the number of produced cells from the first meiotic division. Each produced cell is called a gamete that contains half the number of chromosomes of the parent cell. Each cell of the two cells resulted from the first meiotic cell division is divided in a way similar to the mitotic cell division. So, the cells resulted from the first meiotic division are divided in a way similar to the mitotic division to form 4 cells contain half the number of chromosomes at the end of the second meiotic division. How does the second meiotic division takes place? The two cells which are resulted from the first meiotic undergo prophase 2. In the prophase 2, the nuclear membrane and the nucleolus disappear. The chromosomes are floating on the cytoplasm of the cell. Then the centrosome migrates to the cell spool to form the spindle fibers. Then the cell undergo metaphase 2. In metaphase 2, the chromosomes are arranged in the cell equator of the cell and the spindle fibers connected to the centromere of each chromosome. Then the cell transfers to the anaphase 2. During this stage, the spindle fibers shrink to split the chromosomes into two identical chromatids. Each set of chromatids migrate to the cell pool to form the nucleus. Then the cell transfers to the telophase 2, forming two identical cells to that produced from the first meiotic division. So the result of the meiotic division are 4 cells are produced and each of them contains half the number of chromosomes in the parent cells, which is called the gametes. There are a very important phenomena occur during the prophase 1 in the first meiotic, which is known as the crossing over phenomena. What is meant by the crossing over phenomena? The crossing over phenomena is a phenomenon that takes place at the end of the prophase 1, where some parts of the two inner chromatids of each tetrad are exchanged to produce new genetic arrangement. As shown in the figure, two parts of the chromatids of the inner chromatids are exchanged during their connection to form tetrads in the first meiotic division in the first prophase. What is the aim of this phenomena? This phenomena aims to occur variation of the genetic traits between members of some species, to form variation of the genetic traits between members of some species, which leads to formation and appearance of new traits that doesn't appear in the parent before. Let us see this video to know how does the meiotic division takes place in the reproductive cells. This is the reproductive cells, and this is the centrioles divided and migrate to the cell pool to produce the spindle fibers. The chromatin reticulum intensifies to form chromosomes, consists of two chromatids and the centromere intersected them. Then the chromosomes will connect it together to form the tetrad or the homologous pair. All these occur in the prophase 1 of the first meiotic. This cell is the reproductive one, and this is the tetrad as shown in the video. The structure of tetrad, there are two chromosomes connected together. The crossing over takes place in this stage by exchanging the inner chromatids part, aims to variation in the genetic traits among members of the same species. The spindle fibers are extended to connect to each tetrad. Then the cells is ready to go to the metaphase of the first meiotic. The metaphase 1, the chromosomes are arranged at the cell equator to be connected with the spindle fiber. This is a comparison between the meiosis and mitosis. In mitosis, the spindle fibers are connected to the centromere of each chromosome, but in meiosis, the spindle fibers are connected to the centromeres of the tetrad. Then the cell is ready to go to the metaphase, which is the tetrads are arranged at the cell equator in the same figure. Then the cell enters in anaphase 1. Occur shrinkage of the spindle fibers to split the tetrad into two chromosomes, migrate to the cell pool. So each cell has two sets of chromosomes in each pool. Then it separates in the telophase 1 to form two cells, which each one of the cell contain half number of chromosomes in the parent cells. This is the end of the first meiotic division. Then the cell prepares itself to undergo the second meiotic division. As the chromatin reticulum condenses to form chromosomes, then each chromosome connected to the spindle fibers in the prophase 2, then the chromosomes arranged in the cell equator in metaphase 2, then the spindle fiber shrinkage to form and to split each chromosome into two identical chromatid. Each chromatid migrates to the cell pool to form two sets of chromosomes and forming two identical nucleus. So at the end of the second meiotic, there are 4 cells. These cells are the sperm and ovum. In this part, we will compare between the meiotic division and the mitotic division. This is the meiosis division, where the cell undergo first meiotic division to produce 2 cells contain half number of chromosomes. Then the 2 cells undergo the second meiotic division, which is similar to the mitotic division, to produce 4 cells. Each one of them contain half number of chromosomes, which is called the gametes. But in the mitotic division, the cell undergo 4 stages only, which is prophase, metaphase, anaphase, telophase. And the resulted cells, they are 2 identical cells to the mother cell. But in the meiosis cell division, the resulted cells are 4 cells. Each one contain half number of chromosome in the mother cell. In the meiosis cell division, the cells are haploid, contain N chromosome. But in mitosis cell division, the produced cells are diploid, contain 2N set of chromosomes. Some science technology as nanotechnology and cancer treatment. What is meant by cancer and tumor? What do you know about cancer? Cancer occurs when the body cells are divided continually without any control, and the resulted mass from this division is called tumor. By using the nanotechnology, scientists have developed smart microscopes pumps that penetrate the cancer cells and explode them from inside. They were used to kill the cancer cells in an experimental mice. Mice suffered from cancer were able to live 300 days after this treatment. Other mice that didn't receive this treatment didn't live more than 43 days. The Egyptian scientist Dr. Mustafa al-Sayed discovered a way to detect the cancer cells. This technique starts by loading protein. They have the ability to attach to the cancerous cells. They have the ability to attach to the cancer cell secretions with the nanomolecules of gold. By injecting them to the patient, the infected cell surface and the proteins get intertwined with the golden molecules to make it possible to monitor the infected cells through a microscope, each cell separately. The method of treatment is focusing laser with a certain degree to the gold molecules. Then it will absorb the light and convert it into heat, which leads to the burn and kill all the infected cells that stuck to them. This way of treatment is very successful with the cancer of the skin, and it is applied in the experimental mice and experimental animals. This way of treatment is applied on the experimental animals that show a success in the treatment of the skin cancer. We have to know what's meant by reproduction process. Reproduction process is a characteristic process for the living organism. The living organism reproduces to ensure its existence and increase the number of its species. Again, the living organism reproduces firstly to ensure its existence on the earth. Number two, to increase the number of its species. There are two types of reproduction in our life. Firstly, the asexual reproduction and the sexual reproduction. What's meant by asexual reproduction? The asexual reproduction is the living organisms produce new individuals which have the genetic traits identical to the parents. It occurs in the unicellular living organisms as yeast or amoeba and some multicellular animals as the plants. The asexual reproduction takes place by mitosis division. So, no need for special systems or structures, no change in the genetic structure, where the produced individual is full copy and identical to the parent cell. The daughter cells is identical to the parent cell, contain 2N. The asexual reproduction occurs by only one living organism. The second type of the reproduction, which is the sexual reproduction. The sexual reproduction occurs in the multicellular animals and multicellular plants, or the higher living organisms, where the living organisms produce new individuals have genetic traits differ from their parents. The sexual reproduction takes place by meiosis division, so it needs special systems or special structures. These systems are called the reproductive systems. It occurs changes in the genetic structure, so the new offspring combines the genetic traits from two sources. The first source is the male and the second source is the female. It occurs by two living organisms. One of them is the male and the other is the female. The types of asexual reproduction. The type number one is the binary fission. The binary fission is a type of asexual reproduction where the nucleus divided mitotically, then the cell splits into two identical cells. It occurs in some unicellular organisms as amoeba, paramecium, euglena, some algae, and some bacteria. How the binary fission reproduction takes place. The nucleus divides by mitosis and the cell splits into two cells. Each cell grows and becomes new individual. This figure shows how does the bacterial cell divide. The cell enlarged and the DNA is duplicated. Then the cell wall has to divide and the DNA divided into two identical halves. Then the cell divided into two identical cells. The second type of sexual reproduction is budding. Budding is a type of asexual reproduction, produces new individuals by formation of buds in the parent cell. Occurs in unicellular yeast fungus and multicellular organisms as hydra or sponges. Occurs in unicellular yeast fungus and multicellular organisms as hydra or sponges. At the end of this session, we have discussed together the two types of cell division. The mitotic cell division occurs in the somatic cell, also the meiotic cell division, which occurs in the reproductive cells of the body. See you later and goodbye.